fact, they're usually the other way around. Usually the animal must eat in order to have energy, and in that sense, the animal then has perhaps a surplus of nitrogen and excretes urea and wastes what to a plant would be a fertilizer and a needed food. Vice versa, the plant, on the other hand, then has a great excess of energy, since it can photosynthesize and make use of sunlight, and therefore energy is easily a waste product. Well, it's not been shown that plants make any great use of heat. After all, you don't expect a plant to feel warm to the touch when you go out to the vineyard or whatever. On the other hand, it has been shown they're generally a few degrees warmer than ambient temperature, or a few fractions of a degree. And it might be that uncoupling of oxidative phosphorylation, which would lead to the wasting of the heat that had been stored in ATP or produced by respiration, would be one mechanism that the plant can do something about raising its temperature and possibly doing other useful things in a control sense, regulating how much energy is stored in the form of carbohydrate, how much is released in the form of heat, and how much is coupled into synthesizing energy in the ATP sense. So it's again a fairly obscure and poorly known possible function, but considering that many phenols do have this function, it does appear that it may come to bear on occasion in plant tissue. Then the chelation, solubilization, stabilization, and antioxidant effects are sort of a catch-all related to the fact that phenols commonly, as we've already been discussing, have structures like the bisphenol dihydroxy structures, for instance, which will chelate. We've already seen how borate will chelate with that structure. Other metals may associate in such a way. You could visualize, for instance, if we looked at it, I'm not saying this usually happens, but visualizing some such chelation type arrangement with magnesium or other divalent ions might be possible. And in the case of flavonoids, it's been shown that the 5-OH and the 4-keto group can also chelate with metals, metal ions, aluminum and copper particularly. So that, for instance, a strange case of green bacon in baked beans. They were putting bacon strips in baked beans, and they turned green, and they couldn't figure out why. Well, it turned out after some study that they'd cooked the beans in a copper pan, and there was enough copper present so that the quercetin from the beans chelated with the copper and made a green pigment, and then that collected in the fat of the bacon and made the bacon and the baked beans green. Well, that's a kind of an obscure kind of thing, but if you worked for Campbell's and were selling baked beans, you wouldn't think that was such a minor problem, because people wouldn't buy your green bacon and the baked beans. So that this sort of thing does crop up occasionally, and whether it has useful effect in the plants is open to question, but it is known that plant pigments, for instance, do form lakes. Many of the blue pigments of corn flour and other flours are in fact chelations between aluminum and the pigments, or other metals and the pigments that influence the color and may have some functions other than this. Solubilization and antioxidant are also related to the same thing. Particularly the antioxidant effect is again related to the idea that the compounds are quite readily oxidized, so that not only will they compete for oxygen and in that sense be an antioxidant, but they are also good breakers of free radical chain mechanisms. And remember, we said that the initial product of a one-electron oxidation of a phenol would be a free radical. And the common way of quenching free radical reactions is to have two free radicals meet each other and form a covalent bond by the two unpaired electrons forming a new covalent bond. Well, this is a very common reaction of phenols, and in lipid oxidation in particular, through the formation of hydroperoxides and peroxides, the intermediate. The reason it's a chain reaction is that you have the generation of free radicals, and then as that free free radical reacts with other products, the chain propagates, and you have the autocatalytic effect of the peroxide continuing to catalyze further oxidation. If you have an antioxidant in the form of nordihydroguaretic acid or any other dihydroxyphenol that's active in this regard, it will stop the reaction by combining to make a covalent free radical unit that uses up the free radicals and therefore stops this kind of reaction. Well, there are known protective effects from this, and this relates perhaps to the UV light protection we were talking about. At least it's been shown that irradiation protection is related to this. I mean, gamma ray or other atomic ray type of radiation. You can, for instance, treat rats with quercetin and other such phenolic compounds and then irradiate them with gamma rays at doses that would otherwise be lethal, and a higher percentage will survive if they have a fairly high phenolic intake. So things of this sort, I'm overlapping from plant physiology into animal physiology, but things of this sort do seem to be functions which may be active in plants as well as in the more artificial situations I'm describing. And then the possibility is that the phenols may have a respiratory role, although that seems pretty questionable. No useful role of polyphenol oxidase has been discovered in the sense that it is not coupled with ATP production, so that when polyphenol oxidase produces the oxidation of a phenol, the products do not include useful energy that is trapped as ATP or other high energy useful material. On the other hand, it has an indirect influence in that the total oxygen uptake is certainly influenced, and what respiration is going on would be to a degree competitive, so that the polyphenol oxidase and the substrates phenols may help regulate the partitioning between useful respiration and other types of respiration that don't produce useful energy. Again, it's a little hard to visualize an important physiological role for this, but certainly under some circumstances, such as in tissue cultures where there is a competition for adequate oxygen for normal respiration, then it can influence the net metabolic result, and so it can have a role. The role of phenols as storage substance is pretty questionable as well. It's rather clear that lignin is not ordinarily remetabolized and made use of, so that while starch is obviously an important storage substance, so that, for instance, the grapevine and many other plants store starch in the trunk and roots, so that that is the source of energy in the next spring when the plant begins to grow again. This kind of storage and then reuse as an energy source is not known for lignin, even though it is known that under some circumstances lignin can be metabolized, such things as we've mentioned as a hard seed coat that can be digested and opened so that the sprout can exit from the hard seed. So that even though lignin in rare instances apparently can be remetabolized, it doesn't appear to be a useful energy source. Tannin probably the same, although there is some evidence that under some circumstances, as we've indicated, tannin can be remetabolized, and certainly its energy content would be reasonably high, and therefore some useful energy might result in a plant that was in dark circumstances, say a plant that is grown in the light and has a high tannin content, and then you put it in the dark for an excessive period of time. It's conceivable that its survival might be increased by the conversion of some of the tannin. Again, a rather marginal idea, though, apparently. Well, let's talk about some of these interactions in a little more detail and see if we can get some further sense as to how they operate. First of all, we've already mentioned in specific cases with the grapes, but it's also true with other plants and plant tissues, that the phenolic content can vary in quantitative amount tremendously in a very short time, so that the turnover and production of phenols can be very high, and we'll come back to some examples at the end of this particular discussion. But this rapid turnover certainly does indicate an important role. If they were just to be formed and left alone without possibility of remetabolism, it would be less convincing. There are some environmental factors that influence how much phenol you're likely to find in a plant, and one of them is the carbohydrate-nitrogen balance. Now this carbohydrate-nitrogen, or sometimes called carbon-nitrogen balance, is a rather old concept that modern plant physiologists have tended to begin to poo-poo with justification. It's been very useful in an agricultural, agronomic sense, but doesn't really hang together very well from a physiological sense. Nevertheless, it is a useful concept, and to the extent that it helps explain some of the things that we do observe with phenols is worth mentioning. For instance, it is visualized that there's sort of a balance between the two, so that if you have a high carbohydrate status, this means then that either the plant is inhibited in terms of further growth, or it has reached a stage where it is ceasing growth and is fruiting or going into more advanced physiological condition rather than just rapid growth. And in the maturity sense, you visualize you plant say an annual plant is planted from seed and grows to some degree of size and morphological maturity, and then ceases this growth and devotes its energies, if you put it that way, into producing the fruit or seed for the following generation. Well, generally speaking, if you add high nitrogen, you will delay this conversion. So I may have mentioned earlier, one of the earliest lessons I had in this regard when I was in graduate school, we had a fertilizer analytical group, and they took a pound sample of every fertilizer sold in the state of Indiana and used about 5 grams of it to analyze, and then they had the rest of the pound in a waste bin, and anybody that wanted some fertilizer could get it. Well, raised on a farm where we sweated blood to get enough money to buy fertilizer, I had a little garden and proceeded to bring home probably 100 pounds of fertilizer to put on a garden smaller than this room. Well, the first year I got away with it. I had some of the most beautiful tomatoes you ever saw, but the second year I overdid it. I had so much fertilizer that I had tomato vines as big as this room, but no tomatoes. The blossoms all fell off. The tomatoes grew beautifully but didn't fruit. So I proved to myself that it is possible to have excess nitrogen and not get any fruit. But then this balance does indicate some relationship, and it is interesting that if you do things that increase the carbohydrate status and decrease the nitrogen status, commonly you are increasing the phenol content. So for example, if you girdle, and you know girdling was used to make table grapes larger, for instance. If you girdle the vine, the fruit above the girdle is receiving all the sugar that that cane, the leaves of that cane are producing, and the girdling prevents its migration down into the roots. So you're trapping the carbohydrate in the upper part of that particular cane. You get bigger, sweeter grape berries, and providing the girdle is narrow enough so that it gets eventually bridged and the roots don't die, then you have improved the table grape. Well, under this sort of circumstance, you ordinarily also increase the anthocyanin content if it's a red grape, and if it's not a grape, any other plant, and you do this sort of thing, raising the sugar content by injection, by girdling, by any other means, commonly increases the phenolic content. Even noticing that if you have a peach or a pear with a red side to it, if you look in the tree, the red sides are the ones that are exposed to the light, so that the shady side stays yellow if it's ripe, and the sunny side becomes red. This is even related to the leaves on the tree. The sunny side of the tree, the leaves are commonly higher in phenol, so that this is partially related to light and the synthesis of phenol from light, but it's also partially, or at least indirectly, related to high carbohydrate content. And conversely, sometimes when you have these conditions, you produce deficiency symptoms, including purpling. And if you add nitrogen, the purpling would disappear, so that as you increase growth, you would tend to remove the phenols and lower the phenol content as you do go to the higher nitrogen and more rapid growth condition. Now this may have some relationship to the indole-acetic acid balance, because this is also related to growth rate. If you have high indole-acetic acid or high auxin content, the cells tend to elongate, getting growth, and we'll come back to how that may tie in shortly. Boron deficiency is a rather limited example, but this seems to be tied in at least as an example of a nutritional factor. And I wouldn't say this is completely worked out, but a rather interesting paper by Lee and Aronoff in 1967, and interestingly enough hasn't been followed up by much more work since then. And I'm not quite sure why, but this paper seemed to show rather clearly that in the case of boron deficiency, you have an influence on the phenolic content. Certainly this has been shown that in the case of boron deficiency, commonly the phenol content of the plant does go up considerably. Although, as indicated in your notes, this apparently doesn't happen in corn. It has been shown in many other plants. Well, Lee and Aronoff indicated that borate complexed with 6-phosphogluconic acid. And this borate complex inhibited the 6-phosphogluconate dehydrogenase, and this in turn helped regulate the partitioning of the carbohydrate between the ordinary glycolytic type breakdown and the pentose shunt pathway. Now, if you're increasing the pentose shunt pathway, you should increase the shikimate derivatives, you see, and this would help explain the high production of cinnamate derivatives in boron deficient plants. And not only that, but they further postulated that and showed that the caffeic acid type compound would further complex borate, so that the point is you were already low in borate, and this shifted the metabolism toward the production of phenol, and then the phenol high content would complex some borate by the borate complexing mechanism we've already indicated, and as a consequence you would get sort of an autocatalytic enhancement of the boron deficiency condition. And this even got to the point where the high phenol content appeared to be toxic to the plant, and you got necrosis and death of the plant cells, apparently due to the excessive production of the phenolic derivatives. This has a tie-in also with indole-acetic acid, apparently, in that there is an indication some of the boron toxicity and deficiency symptoms in grapes resemble auxin damage, such things as 2,4-D damage. You know that 2,4-D damage on a grapevine leads to sort of a witch's broom proliferation of growing points and distorted leaves and so on. Well, these kinds of symptoms are present in boron toxicity and deficiency, so that the deficiency tends to lead to the death of the growing point and then a number of shoots coming from it, in the grapevine at least, and the boron toxicity leads to a cupping of the leaf, apparently to inhibition of growth at the margins and enhanced growth in the middle of the leaf, both of which haven't been shown to relate to indole-acetic acid content, but indicate that they may be. Chelation of metals has been mentioned, and metals are involved in the metabolism of phenols, because many of the phenol enzymes require as cofactors some of the transition metal elements like manganese, copper, and iron. And for that matter, of course, phenol oxidase, polyphenol oxidase, is a copper-containing protein. Peroxidases are iron-containing heme proteins, and cobalt, manganese, and others have been shown to be required cofactors. Again, in detail, this isn't known as to why and how specifically this works in some cases. Nevertheless, it would appear that the phenol metabolism would be involved, and deficiencies of these elements would affect phenol metabolism. Less obvious connection relates to potassium and phosphorus, and a deficiency of these elements often gives a change in phenol content. Particularly potassium has been shown to often give high phenol, so that, for instance, if you grow potatoes on potassium deficient soils, then you get a higher phenol content, and this in turn leads to a greater tendency to brown, and that in turn leads to the fact that these potatoes don't make good potato chips, so that potato chips are found to require good potassium fertilization, and for apparently that reason that the phenol content gets too high otherwise. The moisture relationships are perhaps even more directly intriguing. There have been some what I would call gross relationships studied. For instance, a study in Russia where they fertilized, quote, fertilized grapevines with potassium sulfate. Now, potassium sulfate is a good salt substitute for the human diet. Even, well, I don't know about good, but it is used. If you can't take sodium chloride in your diet, they use sodium sulfate as a salt substitute. It tastes salty, it's very water soluble, and so on. Well, if you're going to fertilize a grapevine with potassium sulfate, you almost as well might give it sodium chloride, because what you're doing is you're treating it with salt. You're probably drawing the moisture out of the vine, and this study in Russia indicated that there was a great variability in susceptibility to damage from direct application to crystalline potassium sulfate to the surface of the soil immediately near the vine. Well, further studies indicated that the resistant ones were generally the ones high in tannin. So again, it would appear that the high tannin content had a fairly non-specific effect in this case to resist the depletion of the water supply in the plant. And while this would be non-specific, it might be important in certain kinds of survival, and it's also intriguing that some plants like mangroves, for instance, that'll grow in seawater, at least brackish water at the swamps at the edge of the sea, they are generally very high tannin plants. So mangrove tannin is one source of tannin for leather making and so on. However, a much more specific response has been shown by Willis Gortner and co-workers at the Pineapple Research Institute in Hawaii when I was there. I didn't work directly with them, but I was in the same lab and enjoyed watching their work. And a paper in 1963, followed by several others in other plants by other people, indicated that there was a fluctuation related to the content of chlorogenic acid derivatives, notably paracumaric acid and ferulic acid, in the growing point in response to soil moisture. So that if you plotted soil moisture and you had something above the wilting point and then went below the wilting point and then came back, and you realize on soil moisture, as long as you're above the wilting point, it could be anywhere. Then they found that this was an inverse relationship, so you had a normal level of the ferulic acid, say, and then as the soil moisture went down, the ferulic acid went up, and then as the soil moisture recovered, so did the ferulic acid, although sometimes the recovery was less than complete. In other words, the higher level did maintain for a period of time afterward. Well, whether this has any real significance, it hasn't been possible to indicate. On the other hand, this did occur just in the growing point. You had to trim out the mature tissues and work only with the dividing tissue in the meristematic growing point in order to show this effect. Well, surely the meristematic tissue is the one that is most affected by the drought conditions. You know, the mature cell can hibernate, so to speak, for a period of time, but whether you're going to get increased or growth or not would certainly be highly influenced by availability of moisture. And the fact that this responded very rapidly, in fact, was and has been used in the pineapple as a good measure of soil. You don't have to put gypsum blocks and hygrothermographs and whatever in the soil to measure whether the pineapple needs irrigation or not. You can find it out by analyzing the pineapple. So that it does appear there's some physiological importance. Just why this is done, what's the metabolic benefit of the ferulic acid increase is unknown, hard to speculate upon at this stage. It is interesting, well, perhaps I better, I guess I won't take any further time to enlarge upon the pineapple plant and its great interest. Well, I might take one other point. The pineapple is particularly interesting in this regard because a pineapple is a bromeliad, and you may not realize what that means, but the bromeliads commonly are often air plants, or at least grow on or in trees, for example. And Spanish moss, for example, of the southern states is a fairly close relative of the pineapple, although you certainly wouldn't think so by looking at it. And I've been in pineapple fields, in fact, where you could just walk out and pick up a pineapple plant and carry it away. It just wasn't even fastened to the soil. Now, this isn't the best condition for growing pineapple, you understand, but the plant has a very good adaptation to this sort of thing. For example, in the leaf axil, each pineapple leaf, there's a little root in the bottom of the axil. And the leaf is shaped just like a rain gutter, so if you get any dew and it runs down into this axil, that root is able to absorb it, whereas most plants have no absorptive mechanism and they can't take advantage of dew. So as a consequence, they commonly fertilize pineapple plants by spraying the liquid fertilizer, properly diluted, right on the leaves. And that's why a plant can be reasonably happy without any attachment to the soil. Well, it might be reasonable that such a plant has a very quick responding mechanism to respond to moisture, you see. It's adapted to taking tiny amounts of moisture from dew without rain and growing in, say, in a tree crotch where it would be unlikely to get moisture except during rains. It would have to have some quick response to this condition, and might be one reason it's specially adapted in this regard, or may be specially adapted. Well, other examples are a little less clear-cut. For instance, if you have flooded soil, particularly in winter soils, the shikimic acid content tends to be high in iris roots and low in the dry or summer conditions. And again, this is suggested as a shift back and forth between the pentose path and the glycolytic type carbohydrate breakdown. If you deprive etiolated pea seedlings of water, you get increased indolacetic acid oxidase. And if you have increased indolacetic acid oxidase, you have a lower IAA content, meaning that you're shifting the plant away from growing toward maturing. And again, this would be a natural response. If you're running out of water, you better hurry and make seeds before you die. It would be an anthropomorphic reaction that I think would be reasonable for a plant to take. Temperature, we've indicated, is an important observation. The phenols tending to be high when the temperature is cool. We know this from grapes and wine, although I would point out to you that because the hot country grapes tend to be picked at a higher Brix, and then this produces more alcohol, and if you ferment the grapes on the pumice, you may get a higher tannin in the wine, even though the grapes originally didn't have any more or even less tannin. So if you go through, as I have done, a lot of analyses of wines, you can't always show this effect. And if you look up in the book with Esau, you'll see some examples of this. The reason you can't show it is if you get higher sugar and therefore higher alcohol, you do a better job of extracting the tannin, so that even though there was less there in the first place, you may get more in the wine. But if you correct for this kind of thing, then there is a tremendous difference in total phenol content as well as anthocyanin content in favor of the cool-grown grape as opposed to the warm climate-grown grape of the same variety. And in tobacco, for example, if the tobacco plants were grown at 24, they had 50% more total phenol than if they were grown at 32 when light was controlled to be the same foot candles and so on. So that it does seem to be a general reaction in plants, as not only grapes and the kinds we're interested in directly. Well, 150% of the total is the same as 50% more, right? So I said it the other way, but it's right. In other words, it had more at the cooler temperature. In potatoes, where they're harvested potatoes stored in the cellar, you'll increase the chlorogenic acid if you store them a month at 5 degrees, and if you return them to 20 degrees, it doesn't fall. So that there seems to be produced, and then the degradative mechanism in this tuber at least doesn't seem to exist, and the amount you see is almost threefold increase. Now, if you were measuring normal physiological effects in a plant with a cold-blooded system, you would expect normal reaction rates to be subject to doubling as you increase the temperature 10 degrees. In other words, any synthetic reaction you would expect to produce twice as much product in a given length of time if it was done 10 degrees centigrade warmer. Well, here you're going opposite of that. You're producing more product at a cooler temperature, which makes it even more interesting. Now, of course, sugar level is one factor, and that may be an important factor in the potato storage, for example, because it is true that potatoes stored too near freezing produce the starch breaks down to sugar. So again, it might be the carbohydrate-nitrogen balance type thing that's influencing this, but it isn't just that. And of course, cold hardiness, as we mentioned in the case of salt, may be related to high tannin, which in turn makes the freezing point of the fluid in the plant at a lower temperature. So dissolving anything in the plant sap that is a very good water binder would lower, well, anything dissolved in it, in fact, would lower the freezing point, make it less subject to freezing damage. And certainly, again, sugar content is a more obvious way to do this, but also it apparently is related in some instances to higher tannin content. For reasons that are obvious, the Russians have been busily looking for plants that will grow better at cold temperatures, and their methods of screening have centered on phenols as one, maybe not the best one, but one possible measure, so that oftentimes if you screen for high phenol content, you will then find you have also incidentally produced additional cold hardiness. Mechanical stimulation in response to phenols is rather interesting. There's three components to the story. One has to do with the response to injury. So if you mechanically stimulate with an axe, why then of course you're doing something a little bit more violent, and we'll pick up that under plant pathological studies. But it is sufficient to say that if you injure a plant, commonly there is a response to that injury at the site of the injury or very near to it to produce extra phenol. And we've given examples of the accumulation of phenol, these kinoveins we've mentioned before, or kinoveins. The twining is a rather specific and interesting story that hasn't been well studied, and I keep intending to study it in grapevines, but the period of active tendrils always comes about the time of finals and the spring quarter, and it seems that I never get out and work with grape tendrils at the right time. But pea tendrils have been studied, and it's been shown that the twining reaction is correlated with, now whether it's cause and effect is another question, but it's correlated with a shift between the camphorol and quercetin derivatives in the tendril, so that quercetin is high in the straight tendril, and then as the tendril begins to coil around its chosen support, the quercetin content drops sharply. And then after the coiling is finished and the growth is firm in the coiled condition, the camphorol again tends to rise. And not only that, but this is somewhat light controlled for reasons we'll mention shortly, so that one wonders if maybe it's the light effect more than the mechanical effect. So that, for instance, if you have a mechanical support here and a tendril is growing by, well, obviously it's going to be shaded on the side next to it. Now, if the growth is inhibited in the shaded condition, that's going to cause the outside to grow faster than the inside, which would naturally tend to make it grow around the support. So that light involvement in this case may also be an important function. And as you will be seeing shortly, the diphenolic compound, like quercetin, is an inhibitor of indole acetic acid oxidase. Now, if you inhibit indole acetic acid oxidase, then you're liberating or raising the level of indole acetic acid. This, in turn, leads to extension, so that the balance between monophenol, which promotes the oxidase and therefore inhibits extension, and the diphenol then, which promotes extension, might well have something to do with the twining. So what I'd like to do is not only show that the work with the peas, they just cut off the tendrils and analyze them at various stages. what I'd like to show, and it'd be a little easier, the tendrils of grapes are a little larger diameter than those in peas. I'd like to split it. If this is the tendril, I'd like to take a look at the bottom side versus the top side and see if there isn't a difference in the balance of camphor quercetin, or at least monophenol diphenol, and this may in turn help explain why it's growing faster and curling around the support. In any case, it does suggest a fairly direct control type mechanism. We've already mentioned, I think, that the motor cells in plants that respond to environment, and there are a number of responses. The mimosa is a response to mechanical stimulation. If you step on it or touch it, the leaves will close up. On the other hand, there are quite many other mechanicals or cells that behave in a mechanical way. For instance, there's a tree that grows in Hawaii called the monkey pod. You've seen these monkey pod bowls, for instance. The tree closes the leaves up at night. The leaves are pinnately compound and exposed to the sun all day, and as the sun goes down, they droop and fold together. For what reason, I'm not entirely sure, but in any case, they do this every evening, and this is again the turgor that holds them out straight versus the flaccidity that lets them collapse. Seems to be related to specialized motor cells that produce the movement, and they're the same kind of cells that respond to the mechanical stimulation in other plants, and they are invariably, in the cases studied at least, high in tannin. Now, just what this has to do with it is not entirely indicated, but it does appear that the flaccidity and the turgor balance is governed, just as it is to some degree in humans, by potassium content. And if you make the membrane permeable to potassium, then it becomes flaccid. If you make it impermeable to potassium, it becomes turgid. And the tannin seems to have a role in governing the potassium-water balance that regulates the whole system. To give you one other somewhat related example, I mentioned the mangrove. I'm not so sure about it, but tamarix is another plant tree that'll grow in the brackish sea flats around the ocean. And these plants are found to have salt glands or salt excretion mechanisms, so they can take salt in and excrete it back out. And the specialized glands or cells that secrete the salt back are also very high in tannin content. So these multiple suggestions that tannin has some important relationships in water balance in a specific cell sense and also in a general sense seem to be pretty convincing, even though individually they're somewhat hard to pin down. In total, they seem rather convincing. Responses to light are of two general kinds and mostly related to synthetic, biosynthetic production of phenols. The two kinds of, well, maybe even you might say three levels. There is this carbohydrate-nitrogen balance, and of course if we have high photosynthesis, we're producing high sugar and therefore would tend to produce also high phenol. So that might be indirect. We've also mentioned the sunscreen type effects 2 or 3 times, so that this is another possible relationship where the result is the protection from light or the response to light is somewhat indirect, but does appear to be real. But direct involvement involves the synthesis of phenols and the enzymes that make the phenols, and this seems to be of two general types. There's a high energy reaction and a phytochrome mediated reaction. The high energy reaction is related, is related to the fact that the more light you have, the more phenol you'll make within some limits. And generally it is blue light, which is the most energy containing. The amount of energy per photon of light is greater at the blue end of the spectrum than it is at the red end of the spectrum. So that your high UV and or blue light irradiation gives this high energy reaction. And it seems to have several components that are related to increased phenol. For instance, it seems to stimulate the acetate pathway particularly. And this means that, or perhaps explains the general observation that you tend to have flavonoids in the highest content in, say, leaves at the end of the day, since you're stimulating the acid pathway by the high sunlight, and then cinnamates in proportion tend to be high at the end of the night. So it appears the shikimate pathway may continue in the dark, and then the acetate coupling is stimulated by the high light in the daytime, so that this slight change in relative ratios on a diurnal basis seems to be related to this. There is some suggestion from the action spectra of the particular effects that the enzyme is a riboflavin-coupled enzyme, or a cofactor of a riboflavin component. And acyl-CoA dehydrogenase is known to have riboflavin cofactor involvement. Therefore, again, the acetate pathway, or at least the linking of the shikimate acyl group with the acetyl groups, might be indicated. On the other hand, PAL, phenylalanine ammonia lyase, is greatly stimulated by light, but this tends to be more of the triggering type reaction of the phytochrome type. Triggering in the sense that phytochrome mediated reactions are generally started or stopped by which kind of light you receive last, either red or far red light, and particularly light of 660 or 730 nanometers wavelength. And if you have received the far red light last, then this is absorbed by the particular kind of phytochrome that absorbs 730 light and converted back to the kind that is then receptive to the 660 light, so that you convert the phytochrome back and forth depending upon which kind of light it had last, and this helps control certain reactions of plants that are initiated by different effects. For instance, day length response. And this is apparently related to the fact that the last light you'd get in the evening would be far red light, because this is infrared heat type effect, and you would receive that after the visible red or the more red was gone. So that if this would measure the day length, then would be the length of time that you had red light not followed by far red light. And then the daylight response, which contributes certain plants, converts them from the vegetative condition to the fruiting condition, and so on, would be mediated. And I'm sure you've heard a good deal about phytochrome in some of your other, or at least you should have, in some other viticulture or botany-oriented classes. Well, this has some relationship to phenols as well, then, because the initiation of PAL formation is light responsive, and it does seem to be at least partially controlled by the phytochrome balance. And so that the production of the PAL is initiated by red light, and if you don't wait too long, is reversed by far red light. Now, of course, once an enzyme is produced, the enzyme that's already produced can continue to operate, so that's why it escapes control. You can't flash lights, red, far red, and so on. If you leave the red on long enough, then the enzyme production is already initiated, and you can't turn it off by the far red illumination. Phytochrome of the type that absorbs far red light seems to promote flavonoid synthesis, and the response seems to be, at least in the initial stages, very similar to other phytochrome-mediated responses like day length response and so forth. Interestingly enough, and we've already implied this in some of our earlier conversation, the 3,4-dihydroxy type phenols, the quercetin, etc. type, seems to be generally affected by light more so and stimulated, so that they tend to be produced in proportion at a higher amount in light than the monohydroxy or 4-hydroxyflavonoids. So that, in other words, the light tends to influence the balance between the dihydroxy and the monohydroxy components. Now, that's not entirely so, because in some situations, like say in the anthocyanins, well, we don't have the situation in grapes because we don't have any pelargonidin. and the balance between dihydroxy and trihydroxy is a separate situation. But and we did indicate that the genetics have more to do with setting the pigment composition in grapes than does the environment. But in a more direct sense, like say paracoumaric acid versus caffeic in the more initial stages of biosynthesis, this does seem to be light related, and a feedback mechanism is indicated, because if you add scopoletin, for example, or certain other phenols, then you do inhibit the further production of not only the phenolic products, but the enzymes that produce them. So there is the products of the phenol, like cinnamic acid derivatives, do inhibit the production of phenylalanine ammonia lyase, and that in turn is influenced by light. So that there is a rather complicated but apparently fairly tight control mechanism that does control not only whether phenols are produced, but also which kind in terms of dihydroxy versus monohydroxy at least. We will continue from this point and bring in the auxin relationship. If you haven't already read that, I would suggest you do, because it may be a little hard to follow just from me telling you what goes on. And we will finish then next time the physiology and plant pathological discussion, and then we'll have, of course, our